Now let us discuss the third unit of paper 1. That third unit has two different sections. One is quantitative structure activity relationship, that is QSAR, and another part is the computer aided. The second part is actually this is computer aided drug design in short form, that is CADD. Now uh, in in this section mainly we will focus on the assistance which can be uh, taken from varieties of programs of computer in order to discover a new drug. So in the drug discovery there are many challenges because every day we can't discover the drug, and in this present situation that all over the world people are scientists are searching the suitable medicine for against coronavirus and also suitable vaccine. But as you have seen, the reality is with a full potential also there is no as such much victory. So what are the challenges of drug discovery? Number one is, you know, that for everything price is increasing, so there is a I mean finance matters. Number two is time. See, now you can take the real example of present scenario that badly the whole world needs one medicine against coronavirus or vaccine, but we are not getting. Time is going up, and you know there is a huge competition because whoever will be able to launch the first molecule, that company will get lot of profit, and also the mm, uh, that right to sell in the market. Now, computer aided drug design in a branch of computational chemistry means it is something related to the computer. So, computational chemistry to discover, design, enhance, or study of drugs and related biologically active compounds means it may not uh, be related to the direct discovery of the drug, but it will help us in this journey definitely by, because you can understand now our body is very complex, so simply by using the computer we cannot be able to discover the drug in reality, but definitely computer aided drug design uh, mm, can solve some problems like decreasing the cost, saving the time, uh, like that. So so nowadays computer aided drug design is very important, and I mean preliminary discussion we will be having in this section of this unit, that how computer can help to discover, design the drug or biologically active molecules. So after selecting the disease, see the disease actually that we are selecting. First we are selecting means the company people or the who are actively academic research who are doing, they have to first select the disease, whether it is diabetes, cancer, AIDS, coronavirus, those or that is they will try to discover the cardiac, I mean acting on heart, acting on kidney, or acting on lungs. So first we have to select the disease, and then after selecting the disease, that computer aided drug design comes into the, so that is the first we have to identify the target identification. So the medicine will interact with which target inside our body to control that disease, because you know that for antihypertensive drugs we have discussed already in the paper 2, there are various ways are there to control the hypertension. So first we have to select the disease, and then we have to select that target. So in target identification, bioinformatics, and the three-dimensional structure of the target and the docking study. Docking means by using the computer program, we will see that how the molecule is interacting with the binding site or active site or other site, how the drug and the target is interacting, that is called as docking. So we can take into consideration all these three factors for the target identification. And then when the target identification is over, then we can see the uh, mm, lead identification or the lead discovery, means which molecule is showing the some activity, or it may be very preliminary level, but then also that is called lead, can be modified in the later. So next is the lead optimization. So in order to optimize the lead, that the first part of this chapter means quantitative structure activity relationship can help us. Obviously that is also a part of that is also a computational chemistry, means QSAR also uses some uh, software loaded in computer. And after lead optimization, then if it shows a good uh, mm, uh, mm, that is the pharmacokinetics and pharmacodynamics of that molecule, if it is encouraging, then that molecule will enter into the preclinical trials. That Jyoti madam's paper, unit 1 key, we have all these preclinical trials, clinical trials. So preclinical trials means toxicity study, study on animals and other things, that bioavailability, all these things can be studied. And if everything is okay, then after that we have to do the clinical trials, and their computer aided drug design will not work. So there are varieties of steps are there, stages are there for where the computer aided drug design can be useful. Now, what is the objective of computer aided drug design? Now, to change from the random screening to earlier, when the initially we have all the examples of serendipity, I mean that penicillin discovery by chance, all were there. There was basically no logic actually. So by chance or by random screening means enough number of compounds, if you check the biological activity, then if it shows activity like that, actually initially people used to do like that only drug discovery. But now by taking the computer aided drug design, we can do the rational drug design, means what is the logic between the interaction between target and the drug structure. Number 2 is the speed up the screening process, means if we do logically and if we take the help of computer, then few years can be cut from the whole journey. And then that is efficient screening also we can do. Instead of random screening, we can do the focused screening, and which are not doing any result. Those things we can, instead of prolonging them, carrying them for the different next step, we can remove them if they are really hopeless. So there are four things. Number one is the rational drug design. Number two is the speeding up the screening process. Number three is the efficient screening, focused screening, and number four is removal of the hopeless molecule as early, once it is detected that it is of no use. Now what is, so this is the objective of the computer aided drug design. And now let us see the what is the difference between the computer aided drug design and the traditional drug design. Now see the lead generation or the lead screening, or getting some molecule which is showing the interest against the some particular disease. So we can take the we can do for such type of molecules that biological testing. Now, if we come to the right hand side of this, if it is promising, then we can go for the preclinical studies, and if preclinical studies everything is okay, then that molecule can go to the clinical studies. Otherwise, that is, if we can synthesize the new compounds by getting the by modifying and all these things, and then we can do the biological testing, and so drug design cycle will continue like that. But what is that modern drug discovery by taking the help of the computer aided drug design is, first we have to select the target, and we have to select the ligand, means we have to select the lead molecule by screening already present in the bank of the company or somewhere, or it is a free bank of molecules, otherwise from the natural source. So it can, that natural means it not necessary that it is it is in the it can be under sea also. We in search of new molecules, we can go under the sea also and try to we can exploit that world also, marine world. Now, after understanding the target and selecting the lead molecule, we have to understand the how these are interacting. Now, what is the 3D structure of the target, and the how the when the ligand or means lead is interacting with the active site or the with the target, then how it is interacting, and that is, if we do that thing, that study by taking the help of the computer software, then that study is called as the docking. So we have to do the docking. So I repeat what we do: the rational drug design under the heading of the computer aided drug design. First we have to select the disease, that is okay without computer. Now we have to select the target, we have to select the lead molecule, and then we have to find the 3D structure of the target, and then drug target interaction by docking studies. Now we have to do the structure analysis and the computer design we can do, and then we can modify if it is required. So we can synthesize the structure analysis, we can do which structure activity relationship, means that is that only structure activity relationship, and based on that we can synthesize the new molecules, and then we can do the biological testing, and depending on the moderate activity or the below activity, we can synthesize the new compounds, and if it is promising, then it will go to the preclinical studies, and if it qualifies that round with preclinical studies round, then that molecule will enter into the clinical study. So that is the difference between the traditional drug discovery and the modern drug discovery based on the computer aided drug design. Now, what do we mean by the, now come to this? There are some mm, definitions are there. So let us see what are the definitions here. Now, what do we mean by the active site? Active site is the region of an enzyme where substrate molecules bind and undergo chemical reactions. Now, active site is the whole molecule is not the active, it will be having a specific region, and the 10 to 20% of the overall size of the enzyme is the active site. And as you know, it is the most important part, and because of that only it shows the enzymatic activity when substrate comes and interacts with the active site. Now, active sites can be of two types: one is the binding site and another one is the catalytic site. So binding site means amino acids of the site bind to specific substrate to form the enzyme substrate complex, and the interaction that is called as the binding site. So that is, it can be the, so what could be the interaction that amino acids can participate in hydrogen bonding, it can participate in van der Waals interaction, it can participate in the hydrophobic interaction, or it can participate in electrostatic force. So these are the non-covalent interactions between the amino acids present in the binding site and the substrate. Now another part is the catalytic side. So once the substrate is oriented properly in the active site, then the catalysis starts. So this is actually the what type of catalysis it can mobilize. So one is the covalent catalysis, another one is the acid base catalysis. So substrate forms covalent bond first and then reaction occurs. If nucleophilic catalysis means amino acid is nucleophile, electrophilic catalysis means amino acid is electrophile. Other than this, you will be having the acid base. So there are two types. So that is the covalent catalysis possible, otherwise the acid base catalysis is possible. So for metalloenzyme, metals have very important role during the chemical reaction. Now, other than all these things, we will be having one more site that is called as the allosteric binding site. Now, some enzymes have two sites. One is for the substrate and another is for the inhibitor or the activator. So that is the active site and another one is that is for the substrate and another is for the allosteric site, which is also called as a non-competitive site. Actually, that is for the inhibitor or the activator. So if small molecule comes there, that activation happens, otherwise the small molecules come there, their inhibition happens. So that allosteric site can be used for the activation of the normal reaction, otherwise for the inhibition of some reaction. It can be having the excitatory activity, otherwise the inhibitory activity. Now let us go to, so these are all active sites are over. Now I will go to the docking. So docking is a method to find the best matching between the two molecules. That one molecule is a macromolecule, that is the target which is present in our body, and another one is the small molecule, that is the drug. So the best possible interaction, how it happens, that is called, that process is called as the docking. So now that the docking, what it interacts, now those two molecules will interact or not at all. And if it is yes, then what will be the best possible orientation so that delta G will be minimum, the total energy of interaction. Delta G must be negative. So more negative the delta G, better will be the docking, better will be the interaction between the target and the molecule. So that is what is the purpose of docking? Why should we do? Means the docking is very important to understand the interaction between the lead molecule and the target, and it is very important to optimize and modify the structure of lead molecule to get the better result. Now, results of docking can be used to find the new inhibitors for target and design new drugs. Means there is always a chance to modify the delta G value by changing the structure of the lead molecule and to reduce the cost, because it will save a lot of time. Because one drug discovery, it is approximately $10,000 million and it takes 15 years minimum. So it reduces the cost and also it reduces the time. Now let us see that what is the typical workflow for the docking. Docking is very important as a part of computer aided drug design, where the main interaction between target and the small molecule will be studied so that more delta G will be negative. That can be answered, whether this interaction is good or not, that can be reflected in delta G value. More the negative delta G, better will be the interaction between drug and target. Now what is the workflow? Now as we know, there are two things are there. One is that we have to select the target and we have to select the lead molecule or the ligand we have to select. Now that target, we have to do the preparation, some unwanted things and all these things we have to we have to keep ready this target as well as our ligand molecule, means the lead molecule or the small molecule, which have the potentiality to be the target. we have to prepare them to bring into the dock. They have to be prepared for the docking state. Now after preparation of these things, this can be done by the computer, but all detail how to prepare and all these things, those are not there in syllabus. So just we can remember that we have, after selection we have to do preparation, and then we have to do the actual docking, and you have evaluation of the docking result. If docking if you do, then computer will produce some results, and that results, which results are good, which results are bad, or all the results are bad or not, and if the results are good, then which is the best. Those things we have to do by using our knowledge that computer will not do. That selection process means evaluation process can be done by us. If we are more efficient in understanding all these results, better will be the drug discovery. Now let us see the target selection and preparation. And the 3D structure of the target means here we are protein we are taking. We have to down, this is if it is already solved, then it is available in the protein data bank. That is the protein data bank will be having the structure of the protein which is already solved by X-ray, otherwise by the NMR. So that is a free website, and then depending on our target we have to download it and keep it in our computer. Now available structure should be, if any, we have to check thoroughly that the structure key any mistake is there or not, and if mistakes are there we have to repair all these things. And then the target must be biologically stable. All these things we have to verify. That is the preparation for docking. And active site must be identified. We have to know the active site, because as already we have discussed, the for the total volume or total structure only 10 to 20% is the active site, and that is the most important. There only we have to study the interaction. So we have to download the 3D structure. We have to see if any mistakes, errors are there or not. We have to see that molecule is stable or not, and then we have to identify the active site, and then we have to do the docking. Now from the ligand side, ligands can be obtained from the various databases. There are plenty of data banks are there, or like Zinc or PubChem, that can be, or we can sketch it, means by using the ChemDraw, otherwise the ChemSketch, and then we have to see all the bonds logically they are correct or wrong, and then we have to go for the docking with that particular molecule against the target which already we have selected. Now next is, what are the steps of docking, what we do. So the ligand is docked and interactions are checked, means that can be done by the computer added drug design. We need software like that ChemSketch and ChemDraw. These are the software to sketch the structure of the molecules like that. If we want to study the interaction between target and our ligand, then our computer should have all these softwares, so that to study the docking. So by using that software we have to dock the, means we have to put, very simple, dock means we have to put our ligand into the active site of the target, and then it will do all different conformations, it will check, and at the end it will give some dock score. Dock score is nothing but delta G. So more the negative delta G value, better will be the interaction. So from that we have to select the most negative delta G. Now which softwares are there? Most popular softwares for the docking is the Sanjeevani, that was launched by the IIT Delhi, Gold by University of Cambridge, Autodoc by Scripps Research Institute, and the GemDoc by Taiwan NCT University. These are the most popular software programs used for the docking. So Sanjeevani, Gold, Autodoc, GemDoc, and few of them are free. We can download, and if we know how to dock, then we can proceed for the docking studies. Now, what are the applications of docking? In what way it will be useful? At already you, so virtual screening means where the delta G is not good, we can remove all these things as early as possible. So there is no point in considering those molecules whose dock score is very bad. Now, docking with the scoring function can be used to screen large number of molecules from database quickly to know which molecules can bind with the our target. Now next is the drug discovery lead optimization. So docking can be used to identify, I mean that is in which orientation the target will interact with our molecule. So and accordingly we can design or we can improve the structure of our compound. Now, bioremediation, that is protein ligand docking, can be used to predict the pollutants that can be degraded by the enzyme. So, that is, we can use it for the enhancement of the quality of our environment, that which pollutant is degrading by the protein, that enzyme, that can also be studied by docking. Now, what are the types of dockings? Now, there are two types of dockings are there, lock and key docking and the rigid docking. Means the internal geometry of the target and the ligand are kept fixed. That is lock and key. That is from name it is very clear there. So first relative orientations of two molecules are searched with the separately we have to find the orientation or the conformation of lowest energy conformation for the target as well as the lowest energy conformation for our structure, and then we have to dock both of them. So this is the first way to, I mean it will save a lot of time, and that to perform the initial screening of small molecule database. Rigid docking can be done for different rotamers for a compound individually. So one ligand if we take, we can consider all the conformations, and then we can take the protein, and then rigid docking we can do for all the conformations, and what will be the very good dock score, that only we can consider. Now, what is the drawback of this? The drawback of this is here we are only considering the conformational mobility of the small molecule, means the ligand, but we are not considering the conformational mobility of the target. So with this part is, I mean the first part of computer aided drug design is over. We will come back, I mean in the next session. Thank you.